Yes, Steve, you raise your hand.
Sorry, I was just I accidentally did that. I didn't mean to. Sorry.
Well, it is time to start. So we will do just that. All right. Electron transport chain. Coming to you. Let's have a word of prayer and then we'll begin. Lord, thank you for today. Thank you for watching over us. We pray now you'd help us use the time that remains this semester well and wisely. We pray for our Thanksgivings that we will celebrate. You help us to practice Thanksgiving safely, wisely, and graciously. And now may we learn graciously as well. In Jesus' name, amen. I'm a two-fisted drinker today. So remember, in order for electron transport chains to operate and make ATP, they must be embedded in a membrane. That membrane has to be impermeable to hydrogen ions. They have to be arranged from low to high standard biochemical reduction potentials so that each transfer of electrons yields a positive delta E/0. They have to also alternate between those that carry protons and electrons and those that carry just electrons. So the chain goes from complex 1 if the donator of protons is NADH, to ubiquinone, to complex 3, cytochrome C, to complex 4. And if the carrier is reduced flaving, then it'll go to complex 2, to ubiquinol, to complex 3, to cytochrome C, to complex 4. So it now behooves us to look at each of these complexes. So the first complex is the NADH dehydrogenase, also known as the NADH coenzyme Q or NADH ubiquinone oxidoreductase. This is composed of 45 different protein subunits. Of those 45 polypeptide chains, 38 are encoded by the nuclear genome, 7 are encoded by the mitochondrial genome. Part of this protein is embedded in the membrane, another part juts into the mitochondrial matrix. So this protein will oxidize NADH to NAD. Those electrons will move through a series of carriers. Eventually, those electrons will go to ubiquinone, and ubiquinone will be reduced to ubiquinol. There are 3 subunits that will oxidize the NADH. They're given the names NDU, NDU, ND, meaning NADH dehydrogenase. So there's NDUF-V1, NDUF-V2, and NDUF-S1. These 3 subunits actually work to oxidize NADH to NAD. This monstrosity of a complex is integrated very nicely into the intermitochondrial membrane. The protons get transferred to a flavoprotein, but those protons then get extruded to the other side of the membrane because the next carriers are ions and iron sulfur proteins. Not only the topology of the complex 1 will cause the discarded protons to be extruded, but several of the polypeptides that compose complex 1 that are in the membrane actually have a large degree of similarity with various types of proton pumps. So that part of the protein might very well help pump the protons out as well. There is a nice little pocket for the binding of ubiquinone that will take up the electrons. The protons then will come from the mitochondrial matrix, thus depriving the mitochondrial matrix of protons and enriching the intermitochondrial membrane space for protons. And in doing so, the ubiquinone is reduced to ubiquinol. Ubiquinol can then diffuse through the membrane very, very effectively and ferry those reducing equivalents to complex 3. So here is a crystal structure of a complex 1. This is from the yeast Yarrolia lipolytica. And this is from a paper, Science Magazine, 2017. Did I get that right? Something like that. And no, 2015. So this is from Ulrich Brandt's lab. Ulrich Brandt is one of the gurus of macromolecular complexes. He's in Frankfurt. He does a lot of biophysics on complexes. So the piece that's involved with oxidizing the NADH is right here. And the protein, as you can see, contains these series of iron sulfur centers. These iron sulfur centers are eventually going to take the electrons in this fashion here, through here, around here, to there, over there. And then it says Q, and it turns out this area right here, that is the ubiquinone binding center, right there. And these proteins here that embed this in the membrane, this is very, very rich in alpha helices. These proteins have a great deal of homology to proton membrane pumps. And those protons will be extruded from the matrix to the other side of the membrane. And if you take this, flip it upside down, you get here. So you're flaving, you pass all this to a flavine. So you pass it to a flavine, then it goes to a series of iron sulfur centers, eventually going to a ubiquinone binding site, and these protons get extruded across the membrane. The delta E/0 for the transfer of electrons through this complex to ubiquinol is 0.42 volts. That gives a delta G/0 of minus 19 kilocalories per mole of electrons transferred. This makes transfer electrons through this complex quite exergonic.
What was the delta E again?
Minus 19 kilocalories per mole of electrons transferred.
That was delta G, right?
The delta G, yeah.
What was the delta E?
0.42 volts.
Okay, thanks.
Now, so if the mitochondrial if the mitochondrial genome is deficient and you don't have proper complex 1, you have several different pretty severe genetic diseases. So one of these is called the Leigh syndrome. And this is essentially, this is a complex 1 deficiency. And this particular syndrome is one of several, but there's also so-called fatal infantile lactic acidosis. And this is kind of interesting. If you cannot properly send those electrons down the electron transport chain, where do they go? They go to pyruvate and they make lactate. So you make lots of lactate, you have acidosis. This becomes fatal very early in life, as you can see. There is neonatal cardiomyopathy. There is also leukoencephalopathy, something that is called pure myopathy. No idea why it's called pure, but myopathy, of course, the muscles, which are so ATP dependent, start to break down. And then so-called combined hepatopathy and tubulopathy. So the kidney and the liver, well, both of which require plenty of energy in order to operate, just really go south. So these are the types of conditions that result essentially from complex 1 deficiencies. These disorders encompass a very wide degree of heterogeneity and pathological presentation. They're probably dependent upon several different genetic background effects, environmental effects, things like that, that determine what kind of clinical presentation you have. As you can see, not having complex 1 is quite inimical to life and health, to put it mildly. Okay, so the NADH gives us electrons here. It goes through these series of sulfur proteins. Then it goes to the kind of last stop, if you will, the N2. And then the electrons go to the ubiquinone. And it gets reduced to ubiquinol. and that can then go to complex 3. This is the so-called N and the so-called Q portion of the complex 1. The rest are called P sub P, P sub D, so on and so forth. And these are the bits that this is the bit here. And you see here the little snaking path of the electrons through complex 1. All right, complex 2. This is essentially succinate dehydrogenase, or rather, succinate dehydrogenase is a part of complex 2. This goes by the somewhat formidable name of succinate ubiquinone oxidoreductase or something like that. This is composed of 4 different protein subunits. All 4 subunits are integral membrane proteins, and all 4 are encoded by the nuclear genome. Their genes are called SDH, A, B, C, and D. The electron flow for this reaction has a delta E0, E/0 of 0.05 volts, 0.05 volts, or I guess you could say 50 millivolts. And that gives a corresponding delta G/0 of -2 kilocalories per mole of electrons. You can see the crystal structure here. This has been crystallized from several different organisms. So this is going to couple the oxidation of succinate on one side with the reduction of ubiquinone at another site about 40 angstroms away. You see the path of the electrons here. Here's the substrate binding site. The electrons go here to here to here, and then down to here, essentially. You're going to pump protons out this way. Your ubiquinone is here, and the ubiquinone will also have to pick up more protons from the cytosol. You'll pump protons out to the intermembrane, into the intermembrane space. Notice also that cardiolipin, interestingly, is a component structurally of this protein.
So for this one, the protons are moving from the cytoplasm to the inside the mitochondria.
To the matrix of the intermembrane space.
Okay.
So competitive inhibitors like malinate, or even to a lesser degree oxaloacetate are competitive inhibitors of complex 2. There are also other drugs that inhibit the transfer of electrons from the flavine to ubiquinone. Carboxin is one of these, CARBOXIN. Carboxin. Other enzymes also communicate with complex 2. So when we look at beta oxidation, there's an acyl coenzyme A dehydrogenase that also communicates with complex 2. It uses flavine, and it is also linked directly to complex 2. And then, of course, glycerol 3-phosphate dehydrogenase. is also linked to complex 2, which will ferry reducing equivalents from the cytosol to the mitochondrion. And succinate dehydrogenase, therefore, is linked straight into the electron transport chain. So they go through iron sulfur proteins to the ubiquinone. Those iron sulfur proteins, you can see those here and here. Okay, and here are the other enzymes that also shunt their electrons into complex 2. And all rows are going to meet at coenzyme Q. They will meet at ubiquinone. You have reduced ubiquinone to form ubiquinol. So regardless of whether you go through complex 1 via NADH or complex 2 via reduced flavine, you still end up with reduced ubiquinone. And then you go to complex 3. Ubiquinol will transfer its electrons to complex 3. This is a so-called cytochrome BC complex, or the ubiquinone cytochrome C oxidoreductase. Now, because ubiquinol carries protons and electrons, and complex 3 doesn't, those electrons will just be carried and the protons get extruded into the intermembrane space of the mitochondria. Complex 3 is a very big protein. It's a dimer. Each monomer is composed of 11 protein subunits. All of these, except for the cytochrome B, are encoded by the nuclear genome. And the cytochrome C is a peripheral membrane protein. It'll be attached to the outer surface of the inner mitochondrial membrane. The electrons from ubiquinol go through complex 3 to cytochrome C. Cytochrome C is going to be the electron taxi to complex 4. So here is a structure of a monomer of complex 3. So complex 3 has lots of stuff going on. So you have 2 B-type cytochromes, 2 B-type cytochromes. You have a risky iron sulfur protein. The 2 B-type cytochromes are a high potential B-type cytochrome. Its delta E/0 is 0.5 volts, and a low potential B-type cytochrome. Its delta E/0 is minus 0.1 volts. And then there are a host of proteins that are structural in nature. This is a picture of the dimer. You can see this thing is really big. And here's your cytochrome C, essentially sitting out here, waiting to be released to go someplace else. So here are your 2 B-type cytochromes, high and low, little cavern in between here. Here's your structural proteins, lots and lots and lots of alpha helices to hold it in, and your risky iron sulfur proteins here and here. Very, very complex protein. So cytochrome C is attached to this, not permanently. And also in this protein, you have another C-type cytochrome that's called a cytochrome C1. So who among you can tell me the difference between a C-type and B-type cytochrome?
C-type has a,
Isn't B-type cytochrome an integral, but a C can be integral or peripheral, and it's usually peripheral?
Yeah, but the main structural difference is what?
the C-type is got the cysteine linkage. It's linked to a protein.
How about the way the heme is bound to the protein?
Non-covalent interactions.
There you go. B is non-covalent, C is covalent. So for cytochrome C, think covalent. The heme is covalently linked to the polypeptide. With B types, that's not the case.
Wait, so which one's covalently bound?
C type,
Okay, thank you.
C type. All right, so here is a cartoon of our complex 3. And electrons flow from the ubiquinol, via 2 different pathways. In the first case, let's see, pathway 1, the ubiquinol gives its electron to the risky iron sulfur protein, and then to the C-type cytochrome, and then it goes to cytochrome C. So if we draw this here, let's use our pen. So the electrons are going to go here to here to here. That would be the first pathway for the pathway of electrons. Seemingly pretty direct way for things to go. Now, if you do that, you now have a semiquinone radical. That semiquinone radical is going to serve as reductant for a ubiquinone that is bound to the complex 3 that is called Q sub N. Q sub N. Sorry. Q sub P. Q sub P. My bad. Q sub P. Q sub P. Q sub P is down here. And Q sub N is the one up here. So you remember, you have your low potential here, high potential here. All right. So the Q sub P is going to reduce the, oh, blast it. I'm sorry. I got these mixed. Okay, that's wrong. This should be P, this should be N. My fault. Okay, so P, this is the low potential here. This is the high potential B-type cytochrome here. Low, high. Q sub P is reductant for the low potential cytochrome B. That's going to send its electrons to another ubiquinone that is at the second site where you have the high potential here, which will send it down to here. Now, why does it do this? It does this because one electron is going to go this way. So that's pathway 1. The other electron is going to go this way. This is pathway 2. You reduce this ubiquinone here. This is actually N. This is actually P. Now, why does it do this? Well, because in order for this to receive these electrons here, what does this need? This needs a proton from the cytosol. And now this is going to receive another electron from another ubiquinone that binds. This then can be, so this essentially the ubiquinone gets regenerated, but every time this, here it is. Every time this ubiquinone gives up its electron, these two protons get pumped to the outside. So essentially, one of those electrons comes down here, gets recycled. When another one of these binds, it also gets recycled. So, and then these will rebind. Each electron goes across. You pump 2 hydrogen ions out. Why does it do this? It maximizes the amount of protons that it pumps to the outside of the inner mitochondrial membrane. This is called the Q cycle. The Q cycle. So if we were to go from here, this would be 1. Here's Q sub P. Here's Q sub N. Q sub N, I've got some sort of frog in my throat. So the first set of electrons go here. Second set go to Q sub P, which then go here. Then that sends its electrons to here, which then sends which then sends electrons to here. And these give it to another ubiquinone. And if this is going to be reduced, you have to take up another hydrogen ion. So with this transfer, though, you gave it 1 electron, but you're still pumping 2 protons out here. For every one of these that comes up, you're doubling essentially the amount of protons that you pump with each one of these. And I don't know why when I try to write with this, it does that, but that is a common issue with writing on this. Okay, that's a Q cycle. That's a Q cycle. And 2 electrons are given, 2 protons are pumped, but one gets, because cytochrome C can only carry 1 electron. The other electron goes down here and gets recycled. That maximizes the number of protons that you pump to the other side of the membrane. So one electron then goes to cytochrome C. The ubiquinol is regenerated to give its electrons up again. Ultimately, because the thing's a dimer, you'll have 2 reduced ubiquinones that will donate their electrons to complex 3. One will be regenerated, one will be depleted. 2 electrons then will go to 2 cytochrome Cs that will then go to complex 4. Incidentally,
I have a quick question.
yeah,
That last slide said that the 2 electrons go to cytochrome C, but I thought cytochrome C only got 1 electron, or does it do
right.
it
It takes, because it's a dimer. There are 2 cytochrome Cs bound. Each
Okay.
one gets 1 electron. Now there is a mutation in a gene that goes complex 3 that's actually been worked on to some extent. This gene is called BCS1. It encodes a component of complex 3. This gene is very, very heavily conserved in yeast. In fact, that's what it that's where it was first characterized, and mutations will cause this so-called Gracil syndrome. And that is your growth retardation amino aciduria, cholestasis, iron overload, and then early death, and you might have guessed what this one is, lactic acidosis. So without complex 3, you are in a world of hurt, shall we say. There is another genetic disease associated with problems to contact complex 3, and this is the BCS1L gene. So mutations in this will cause something called Bierstadt syndrome. This can also be caused by mutations in the cytochrome B gene as well. So complex 3, mutations in complex 3 significant genes have significant consequences for people unfortunate enough to have them. All right.
So those symptoms, well, all the acronyms for the Gracil syndrome, do they appear all of them at once, or you might have one or the other?
They appear early in development, and they tend to appear gradually. The order in which they appear, the growth retardation, you notice that usually because the growth, that takes time to notice. The lactic acidosis, you start to see that rather early. The aminoaciduria, you see that rather early. Cholestasis, that takes a bit, and so does iron overload. So they tend to arise progressively, and they tend to get progressively worse. Unfortunately, there's little you can do for someone with those types of conditions.
How early death?
Usually before the first year of life, sometimes as early as 6 months. One other feature I forgot to mention. Complex 3 is subject to inhibition by several chemicals. So antimycin blocks the transfer of the electrons from the high potential cytochrome B to ubiquinone. Mixothiazole prevents the transfer of electrons from ubiquinol to the rescue iron sulfur protein, or the P position ubiquinone. And then stigmatelin prevents transfer of electrons from the iron sulfur protein to the C-type cytochrome, and then ultimately to cytochrome C as well. So these three different inhibitors inhibit complex 3. I'll leave that up there for a few shakes of a lamb's tail to let you get those down. Okay. All right, so that Q complex, Q cycle means that you're going to maximize the number of protons you pump out the membrane. Then cytochrome C will play the mobile carrier that sends the electrons from complex 3 to complex 4. And complex 4 is the cytochrome oxidase. This contains 3 atoms of copper and 2 A-type cytochromes. Has a cytochrome A3. The copper undergoes transition from a C copper 1 to a copper 2, so from cuprous to cupric states during the transfer of electrons to molecular oxygen. The 2 copper centers are termed CuA and CuB, copper A and copper B. The copper centers are critical in the reduction of oxidized iron of cytochrome C, which was oxidized by the electrons from complex 3. And the iron 3 of complex of cytochrome C, that's the site of cyanide inhibition of complex 4. So cyanide is quite poisonous. It was used, unfortunately, by the Nazis in World War II in the gas chambers. Zyklon D is essentially hydrogen cyanide. Zyklon B. Complex 4 is composed of 13 protein subunits. Oxygen is the final electron acceptor. And the final product, of course, is water. The catalytic core of this is encoded by the mitochondrial genome in humans, as are the cytochrome A's. There's also a structural protein called CO3 that's encoded by the mitochondrial genome. The other subunits, the other 10 subunits are encoded by the nuclear genome. And 2 of these polypeptides actually have heart-specific and skeletal muscle-specific and liver-specific transcripts that lead to slightly different types of proteins. So here's a crystal structure of these. You can see the copper centers in green, and the heme group is there. There's your copper up here. There's your iron. This is a very beautiful little picture from your book of a electron microscope picture taken of cytochrome oxidase with complex 3. And it seems as though you have 2 different cytochrome oxidases here, and one very large complex 3 that sends electrons essentially to them this way. So these are very tightly associated with each other in the membrane. These structures are often called respirasomes. Respirasomes. So the copper atoms with your cytochromes A and A3, it takes 4 electrons to reduce 1 molecule of oxygen to 2 molecules of water. That shouldn't be too surprising, given that 2 molecules of water will have 4 hydrogens between them. The production of water further removes hydrogen ions from the matrix. This creates a disparity of charge, pH, and concentration on opposite sides of the mitochondrial membrane. So of the 13 subunits that mammals have in the cytochrome oxidase, bacteria have essentially the 3 or 4 core polypeptides in their cytochrome oxidases. And the conservation, amino acid sequence conservation between those core subunits is pretty impressive. As we said, this generates water. And the water is generated after the oxygen binds to the B position, copper. So here's your 2 coppers here that take the electrons from cytochrome C. They then get transferred, they being the electrons, get transferred through the 2 hemes. They go to the B-type copper that binds the molecular oxygen. It gets reduced, it takes these protons here, and then it gives you water. And this is a summation of how this reaction probably occurs. So this is the iron from the cytochrome A3. Here's copper B. So oxygen comes in. You go from copper 2 to copper 1. You're holding the molecular oxygen, the iron goes from iron 3 to iron 2. As you accept electrons, now you go from copper 1 to copper 2. You accept more electrons. You now go to copper 2, iron 4, and then you accept your hydrogens, as you can see here, and we have a water here, and then you have a water here, and you release it essentially that way as water, and then you regenerate your original status here, ready to bind another oxygen. Notice that the iron is bound once again by histidine, just like what you have in hemoglobin and myoglobin, and so on and so forth.
Dr. Baratovich, in that just the previous slide to the other slide, there's 4 hydrogens that bind to form the water, and then it says 4 hydrogens that are pumped. Those are different ones. Why?
That means you have a disparity of 4 protons from either side of the membrane. So it's as though you pumped 4 protons.
Okay, so it's not that you're removing 4 hydrogens to make the water and you're pumping 4 hydrogens.
No,
so it's just
no.
the equivalent of
That's equivalent,
Okay.
the energetic equivalent, as though you pumped 4 protons because you depleted the matrix of 4 protons.
Okay, thanks.
Yeah, that picture is a tad confusing on that one. And this kind of sums the whole thing up. So remember, your ubiquinol carrier from complex 1 and 2 to complex 3. Cytochrome C is a carrier from complex 3 to complex 4. You have the Q cycle in complex 3, and then the electrons go through the 2 copper centers and the A-type cytochrome ions to give electrons to molecular oxygen to make water. And each time you do this, you pump protons across the membrane. You pump the protons across the membrane. Now you enrich the intermembrane space for protons. Very good. So you made water, you pumped all those protons. Now what are you going to do with all those protons? You're going to use them to make ATP. And you're going to make bucket loads of ATP out of this. All right, so the transfer of electrons down the electron transport chain is thermodynamically spontaneous. You have a very negative delta G/0. You have a very positive delta E/0 because you've arranged the carriers in the order of very negative delta G/0s to very positive delta G/0. As I said last time, descending electronegativities, that's not quite right. It's ascending standard biochemical reduction potentials. You go from negative delta E's to positive delta E's. So that transfer is spontaneous, thermodynamically speaking. And then you can see here, you go from NADH, which has a standard biochemical reduction potential of minus 0.32 volts. I didn't put the volts in there, I should have, sorry. To molecular oxygen, whose delta E/0 is plus 0.816. That's pretty spontaneous, thermodynamically speaking. So if you calculate then the delta G/0 using this equation, you can see, you put the numbers in, and you can see the units are going to cancel pretty nicely here. Volts, you're going to get kilojoules per mole. That'd be mole of electrons transferred. Is going to be minus 220 kilojoules per mole of NADH. And that's pretty spontaneous. This gives you a proton gradient on either side of the inner mitochondrial membrane. The inner mitochondrial membrane is impervious to protons. Therefore, you have the potential to do work. So thermodynamically, we have concentration disparity. We also have a pH disparity, and we have a charge disparity. This potential to do work is called a proton motive force. A proton motive force. A proton motive force exists when you have a disparity of protons. That is, you have a differing proton concentration on either side of a biological membrane that is impermeable to protons. Therefore, you have a pH differential, you have a concentration differential, and a charge differential. Each one of those, you add them together, will give you the potential to do work. Now, the cell, the mitochondrion, is going to take these and it's going to move them through the ATPase right here. In doing so, it's going to make ATP. So that's the work it's going to do. It's going to make ATP out of this. That's how it's going to utilize this battery, essentially, that is made with this process of electron transport. So here is the thermodynamic calculation of this. So you have C1 divided C2 divided by C1. That's your proton concentration differential, proton concentration in the matrix relative to the proton concentration in the intermembrane space, plus Z times Faraday's constant times delta of C. These all account for the charge and pH differential. So if we, so the charge Z, that's +1. Delta of C, that's the pH differential. We can measure that. So we have this equation here. These are two constants. We can put those right here. And the proton motive force, we just explained that. Difference in concentration of protons across the membrane. And the delta G is expressed as this. You have concentration difference. You have electrical charge of the ion +1. And the delta of C is that transmembrane difference, the charge difference in electrical potential that's measured in volts. Again, that's something can be measured.
Dr. Bradovich,
Yeah.
how do they decide? How do you decide what C2 and C1 is? Which one is is it arbitrary?
No. This is where you're doing the work. The work that you're going to do is from here to here. So this is the one essentially that you're going to be pumping to, because you want to know how much work essentially you can do through this delta G. How spontaneous it would be to let these in. So therefore, if you want to know what it is to let them in from here to here, this is where you're coming from. This is where you're going to. You have where you're going to divided by where you're coming from.
Okay.
If you want to pump them the other way, then you swap these, because then that's the work you want to know to go from here to here.
I was thinking of the F-type ATPase pump for some reason, but it's a proton pump.
That's, yeah. All right, so this number turns out to be a difference in pH. because that's what a concentration of protons is. So we can then take this thing right here, the logarithm, this here, and just turn it into delta pH. Now we have something a bit simpler to deal with because we've turned that now into the log base 10 instead of log base E. That's why we have to do the 2, 3. And we have 5.7 kilojoules per mole times delta pH plus this number here, because that's a constant, Faraday's constant is a constant, times delta C. And if you want to know the charge differential, you can stick an electrode in an electron. Yes, that's hard to do, but it's been done. Turns out this number is about 0.15 volts to 0.2 volts. And then the pH difference is usually about 0.75 pH units. And so if we solve for delta G, we get about 20 kilojoules per mole of protons. Lots and lots of potential to do work there. So for every mole of protons, your delta G/0 is minus 20 kilojoules per mole. Can you use that to make ATP? The answer is, you bet your life. Okay, how do we get the protons back into the matrix? One of the gentlemen to work on this issue was this guy here. This is Peter Mitchell. Peter Mitchell is the father of the chemiosmotic or Mitchell hypothesis. So Leininger worked and worked and worked his whole life looking for that intermediate, high phosphate intermediate. It was Peter Mitchell who actually took what Leininger did and finished it out. I was in a seminar once with Peter Mitchell, and I think after the first 5 minutes, he lost probably 95% of the crowd that was there. There were 3 professors in the front, I think, who understood what he was saying. And I was busy as he was talking, reviewing my calculus, because most of his talk was calculus. And by the time he got done, he said, "Are there any questions?" And everyone in the room literally laughed because he was just so far above. He was a brilliant man. He really was. Okay, so that's the end of today. Oh, crumbs, I went over time a little bit. So I will see you guys on Monday. We'll talk about the Mitchell hypothesis, and then we'll look at the work of people like John Walker and others, F-type ATPases, and so on and so forth. Have a great weekend, and I'll see you then.
See you Monday.
Bye.